Hello friends, welcome back to my channel Plant Pathology Pathsala. As you all know that we are discussing the plant virology part, the unit 6. So in this series, the plant virology series, we have already discussed means we have already covered 7 video. This is the 8th number video, and in this video we will discuss about symptoms produced by plant viruses. So symptoms means these are the manifestation, the visual manifestation of the microorganism, or you can say the virus on plant, because at cell level how this virus affecting the metabolism as well as cell structure or cell functioning we can't see. What we see, the symptoms we will see, either necrotic symptom or yellowing, curling. These are the symptoms produced by the pathogen that we see visually over the plant, because the main thing is because the virus hampers some metabolic activities of the plant, that's why these symptoms are produced. So most common symptoms produced by plant viruses are the mosaic, yellowing, stunting, leaf curling, then ring spot and rosette. We will discuss about all these type of symptoms one by one. What is mosaic? How it is different from mottling, yellowing, then the chlorosis, intervenal chlorosis, vein banding, then streaking, strips. All these different terms we will discuss all one by one. So we have classified the plant virus symptoms or symptoms produced by plants, symptoms produced by viruses on plant into 4 major categories, that are the macroscopic symptom. Macroscopic symptom means that are large enough to visually inspected by our normal human eye, like the leaf spot or yellowing, mosaic. There are macroscopic symptoms. Then histological symptom. Histological symptom is at tissue level what symptoms virus produce, like hyperplasia, hypoplasia. Okay, we will discuss that. Then third is the cytological symptom. Cytological symptom means cyto means cell. At cellular level. At cellular level means at cell level any changes in the cytoplasm or nucleus. So any cell organelle changes. So these things we will discuss. And final, the most important, inclusion bodies. These are the special structures produced by specific virus. Specific virus only produce some specific particular inclusion bodies. So that we will discuss. So let's start. So symptoms, the macroscopic symptoms are generally 2 types: the local symptom and the systemic symptom. The local symptom are, and again 4 types, that may be necrotic local lesion, or maybe chlorotic, ring spot, or starch lesion. So what is first local lesion? Local means the name only defines that it will produce at or near the entry point of the pathogen or entry point of the virus. So local symptoms are symptom form near the point of virus entry on the leaf, and it is localized, not spread out to the all other part of the plant. At the entry point only, and that symptom will be produced. So definitely it will be small lesions only. So that lesions may be of 4 types. First is the necrotic lesion. Necrosis means cell death. So here small dead spot at the site of infection will be seen, and that dead spot are due to the hypersensitive reaction. Hypersensitive reaction is due to the oxidative burst. This is actually response of plant to the infection by pathogen. Due to the oxidative stress, the cell death or cell necrosis occur. due to the programmed cell death. Okay, this is to restrict the pathogen. So first example of local symptom is necrotic local lesion. Mostly necrotic local lesion producing virus example is wheat yellow virus. Here in the photo you can see here small necrotic lesions are there, and that these lesions are not widespread, these are restricted, these are local symptom. Then the chlorotic lesion. Chlorosis means loss of chlorophyll. So here yellow lesions will be developed due to the chlorophyll loss at or near the entry point of the virus, often surrounded by the green tissue. Example of virus producing chlorotic local lesion is the tobacco mosaic virus, the TMV, on Nicotiana glutinosa. Then the third major local symptom is ring spot lesion. Ring means concentric circle. So here the virus will produce concentric chlorotic as well as necrotic. Chlorotic means yellowish lesions, or necrotic means dead spots. So dead lesion or chlorotic lesion that will be formed in concentric ring pattern. Here in the photo you can see here these rings are formed in a concentric pattern, one over another, one around another. Okay, that may be formed over leaves, fruits or tubers. So example of viruses producing necrotic lesions are tobacco ring spot virus, papaya ring spot virus. So this ring spot are generally produced on fruit, leaves, tuber, at any part. Then the fourth important lesion local symptom is starch lesion. So starch lesion, obviously it is due to the accumulation of starch. And accumulation of starch, generally we observe starch test, how we do? By using iodine staining. So actually for its own metabolism, virus divert the starch accumulation at or near the point of infection, that is resulting in formation of starch lesion. Example of virus producing starch lesion is the TMV, tobacco mosaic virus, on infected tobacco leaves. Here you can see this iodine stained the starch lesions. Okay, next the systemic symptoms. So systemic symptoms include the stunting, yellowing, mosaic, mottle, then vein clearing, vein banding, and then strips, color breaking, leaf curl. So various symptoms are there. So systemic symptom, the first one is stunting. Stunting, or it is also known as dwarfing. So what happened? The plant in stunting, the plant height will reduced, and why? Because of reduction in internodal length. Leaves also become smaller and plant will show poor vigor. This is the most common symptom in most of the viral diseases. This stunting is very common. Stunting and yellowing, these are most common symptoms for plant viruses. Example of virus producing stunting is rice tumor virus, barley yellow drop. This rice tumor is very important. Okay, next yellowing. Yellowing, how this leaf yellowing occur? Because of degradation of chlorophyll. So general chlorosis will occur only due to the degradation of chlorophyll. It may start as vein yellowing and progress to whole leaf. Example is wheat yellow virus, barley yellow drop virus, rice tumor bacilliform virus. This is about the stunting and yellowing. Then about the mosaic and mottle, the most common symptom or most characteristic symptom. So this is very confusing topic actually. So many people confused about what is mottling and what is mosaic. So mosaic means alternate dark and green light areas, and this green and light area are actually distinct Means clear distinction will be there between the light, this alternate dark green and light green areas. This is the most characteristic symptom. And why this occur? Due to uneven development of chloroplast. Wherever any type of yellowing is there or chlorosis is there, definitely that is due to the uneven development or loss of chlorophyll or chloroplast. Example of viruses producing mosaic symptom are TMV and CMV. Then the mottling. Here the alternate dark and light green areas are clearly distinct, whereas here in the mottling case they are less distinct and irregular. It is like diffuse, not clearly distinct areas are there. This diffused chlorosis is there, mild and diffuse chlorosis. Okay, so mottling means mild diffuse chlorosis, whereas mosaic means alternate dark and light green areas. So less contrast between light and dark green areas than mosaic. Example is CMV. Next, the vein symptom. So the systemic symptoms producing on veins are categorized into two types: the vein banding and vein clearing. So vein clearing means the clearing of tissue of the vein. The name itself suggesting this. So the vein tissue become translucent or glassy. This is the earliest symptom and most visible when held against the light, because that area will be transparent when you place the leaf or the surface against the light, then you can clearly see that. So example of viruses producing vein clearing is the tobacco itch virus and CMV. Then the vein banding. So in vein banding, dark or yellow bands along the vein will be there, and band may be continuous or broken, often seen in mosaic and yellowing disease. So example, same CMV and beet yellow viruses. So difference between vein banding and vein clearing is, in the vein clearing, the venal area will become glassy and translucent, whereas in vein banding, the vein area will become more greener as well as yellowish, not translucent or glassy. Next, the strips. So strips means the long chlorotic parallel lines will appear on the leaves. Here you can clearly see the parallel lines will be there. These are called as strips. Long lines. Example are banana streak virus and maize streak virus. These produce the strips symptoms. Then color breaking. Actually, strips and sticks. Strips generally used for dicot plants, and the sticks used for a monocot plant. It's the difference only. In both the cases, the parallel chlorotic lines will be formed. Then the color breaking. Color breaking, this is the tulipomania. This, in history of virology, you might have heard this. This is due to the color breaking of the tulip petals that increase its demand, and the demand is so high that it is sold more than gold, and this is due to the loss of chlorophyll pigment. So irregular streaks or sectors will be formed of different colors in petals. Here you can see here irregular streaks are formed or lines are formed over the petals. This actually enhances the beauty of the flower, but this is not due to any genetic or the plant characteristics. This is due to the virus infection. This is due to the infection of potty virus, that is tulip breaking virus. Okay, that is causing this irregular streak formation. Same also produced by TMV on petunia. Then the leaf curl and leaf roll. Leaf curl and leaf roll, same thing only. Leaf curling means the leaf will be curled down. Why this actually curled? Because of abnormal elongation or abnormal growth of the leaf tissue, as well as some starch accumulation also leads to this curling of the or crinkling or distortion of the leaf. Okay, so this is about the leaf curl. This is the symptom for leaf curl. Sorry, this is the symptom. You can see cupping, cup like formation, crinkling of the leaf. So example of virus producing leaf curl is tomato leaf curl virus, tomato and then cotton leaf curl virus. Then leaf rolling. Leaf curling is abnormal curling or crinkling or distortion of leaf. But leaf rolling means total rolling of leaf, either upward or downward along the margin. Okay, either downward or the upward rolling. Often associated with yellowing and stunting, caused by many viruses, mostly by the viruses that are vectored by the insect pest. Example is potato leaf roll virus. Here you can see the clear rolling of leaf. Next symptom produced by virus. This is a histological symptom. This is a histological symptom only at tissue level. So this is due to the abnormal growth or abnormal tissue growth that is leading to structural changes in the infected plant. That is either due to the hypertrophy or hyperplasia. So example of developmental abnormalities are, first is the ination. This is most important symptom, and ination produced by the virus. This is also important. The P ination mosaic virus. Remember both example as well as the symptom. So what is ination? So ination are outgrowth or warty projection on leaf vein or petioles. You can see here warty projection. So means some outgrowth, abnormal cell proliferation are there on the leaf vein as well as at the petioles of leaf. Here you can see here. Then the swelling. Swelling means abnormal enlargement of stem, shoot, petioles or vein due to the excessive cell division or enlargement. Excessive cell division means hyperplasia. Excessive cell enlargement, hyperplasia. Hypertrophy. So plant will appear thickened and succulent. So example is cocoa swollen shoot virus that produce swollen shoot on cocoa plant. Then the tumor formation. This is also due to hypertrophy and hyperplasia, that is due to uncontrolled cell division, that is leading to formation of tumor and galls. So tumor may occur either on stem, root, or any wounded part. Best example is wound tumor virus. And remember this, the tumor formation generally mostly associated with dicot plants. The tumor virus never, in fact, you have never seen any tumor type of symptom in the monocot plant. This is for the dicot only. Even in the bacteria also, agrobacterium infect dicots only, never infect any monocot. So then other symptoms are, other common symptoms is the rosette, wilting. So rosetting means the leaf will be clustered in a manner that look like rose. Okay, here bushy appearance will be there. That's why the name is rosette, because of rosy appearance. In the rose, the petals are clustered they are very bushy like structure. Just like that, here also the leaf, due to the shortening of the internode, the leaf will be clustered and that look bushy. That's why they are called as rosette. Example of virus producing rosette symptom is groundnut rosette disease or groundnut rosette virus. Then wilting. Wilting, this is due to the collapse of the vascular tissue that obstruct the water flow and resulting in wilted plant. Example of virus producing wilting symptom is tristeza. Citrus tristeza virus. This is a very important virus. Then recovery. Some virus actually A new growth becomes symptomless through virus persistence. Generally, the plant will recover the symptoms, and mostly it is associated with the nepoviruses. Okay. Next, about the histological symptoms. These are the cellular changes or tissue level changes that we have already discussed. These are histological changes only. Okay, and that are classified into two major categories: the hypertrophy and hyperplasia. Hypertrophy is due to the cell enlargement, and hyperplasia is due to increased number of cell division. Already I have discussed. Then the inclusion body, this we will discuss in the cytological part. Okay. So cytological symptom. So cytological symptom means the alternation in cell or their organelles due to viral infection. It may be either the changes associated with nucleus or cytoplasm or any organ like the major organelle, the chloroplast and mitochondria. So nuclear changes, first the nuclear changes. So there may be any changes in the nuclear size, shape or structure of the nucleus. So the example is onion yellow dwarf virus. Then cytoplasmic changes means, and the vacuolation will be there, or disorganization of cytoplasm, or any inclusion bodies may be formed at the cytoplasm. Okay. So example is TMV. Then chloroplast. So some virus also may either increase the chloroplast or may degrade the chloroplast. Degradation of chloroplast leads to the yellowing symptom of chlorosis, but increased number of chloroplasts will lead to the starch accumulation. Example is the CMV. Then mitochondria changes. Some viruses also cause swollen or less functional mitochondria. Example is tomato spotted wilt virus. So this is about the cytological symptom. Then finally, most important is the inclusion bodies. So these are microscopic structures formed in infected plant cell, and this is most important for our perspective for identification of the virus. Actually, what happened during the virus replication, when excess proteins or viral bodies are formed, they are removed as inclusion bodies from the replication pool, and that are forming some crystal structure that are known as inclusion body. And depending on the shape that inclusion bodies are forming, these are actually characteristic shape. This means particular virus will produce particular type of inclusion bodies only. These are diagnostic structure for the viruses, like the crystalline inclusion body. The crystalline means it is a crystal like structure. This is a well defined structure or crystalline array is there. Example of virus producing crystalline inclusion bodies is TMB, tobacco mosaic virus. Remember the name of the crystalline body, the type of crystalline body, as well as the example. This will be directly asked in the exams. Okay. Then about the pinwheel inclusion body. This is the characteristic structure produced by potty virus. Okay. Here you can see in the photo that pinwheel like rosette structure. This pinwheel like structure is there. Okay. This is the clear indication or diagnostic for potty virus. Then cylindrical inclusion. Cylindrical inclusion means cylinder like shaped bodies formed in the cytoplasm by potato virus Y and tomato each virus. Then the last virus inclusion body. These are the granular or amorphous inclusion body produced by the virus, cauliflower mosaic virus. So remember both the crystalline in both the inclusion bodies type as well as the its example also. So these are the diagnostic structure for identification of the virus at microscopic level. So this is about the symptoms produced by a virus, the macroscopic symptom, then the symptoms produced at histological level, then symptom produced at the cytological level, means at the nuclear cytoplasm or chloroplast at or mitochondrial level, as well as finally the most important diagnostic symptoms produced by plant viruses, that is the inclusion bodies. So here we have covered all about the symptoms produced by plant viruses. I hope everything is clear to you. So thanks for watching this video. In the next video, we will definitely discuss about virus transmission and other things. Thank you.